Welcome everyone. I'd just like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. So good afternoon everyone and welcome to the FightMND Scientific Seminar for March. My name is Sarah Bennett. I'm the Research Coordinator at FightMND. Thank you for joining us for the scientific seminar this month. We had a wonderful presentation in February on the use of wearable monitoring in MND care and research by Dr. Derek Stein and Corey Holden from the University of Queensland in February. So now we have bring you an international guest speaker for our March seminar. So following the presentation today, we are leaving plenty of time for discussion and questions. So if anyone would like to ask anything, please do use the reaction button to raise your hand and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, if you don't feel comfortable, you can also enter your question into the chat where our team will be monitoring and can read out the question on your behalf. Please note, however, that the chat function is only enabled if you are using the app-based version of Teams and unfortunately doesn't work in the web-based version. Please ensure that you do introduce yourself and where you're from before asking your question. So for today's presentation, we have a distinguished international guest speaker. Dr. John Ravits is a Professor of Clinical Neurosciences and Head of the ALS programs at the University of California in San Diego. John will be presenting to you his talk, Targeting CK1-mediated TDP-43 phosphorylation in ALS. So John, I have a couple of questions for you, just for our audience to get to know you a little bit. Can you tell us what motivated you to specialize in the ALS field?
Well, I began practicing, at the beginning I was primarily a clinician, so I was seeing the ALS patients, and I guess I was getting kind of frustrated at the lack of progress in research and how difficult it was, and began to have my own research ideas based on onset and progression and what I thought were key features of the disease. And so I ultimately decided that as a clinician, I needed to pursue that because other research scientists and neuropathologists were not able to make the observations that I could in the research arena. And I was trying to drive those forward.
Yeah. So what do you think is your proudest career achievement to date?
Ooh, probably, I think I'm proudest of that, of writing papers that I thought were relevant to the onset and progression of patients with the propagation, anatomical spread of the disease, and writing those to and connect them to basic science of propagation and of molecular mechanisms.
Yeah. Well, bit of a fun question now. If you were to have discovered a gene, and of course called it Ravits1, what do you think its function would have been?
If I could discover a gene for the cause of ALS?
Yeah. Whatever cause you think it might have been.
Yeah.
Ravits1, the sole cause of ALS.
Ravits1 would be I guess it would have to do with propagation. So it would be something that folds and increases itself as a molecular kind of folding mechanism.
Well, thanks for playing along. So over to you now for your presentation. Thank you.
Okay, well, thank you for inviting me and having me, and let me share my screen. There. Are you able to see my screen?
Yeah, that's all good. Thanks. think you just need to full screen.
Here we go.
Yep, that's great. Thank you.
Yeah, are you full screen now?
Yes, thank you.
Thanks.
Okay, great. Well, first of all, I just want to thank FightMND and you all for including me and inviting me to share some of the work that we're doing in my laboratory, and particularly that's been supported and funded by FightMND. So I'm excited to share where we're at with it. I think, spoiler alert, is we are poised with some pretty interesting experiments coming ahead, and I'll share that with you. And hopefully in a year and a half, I'll be able to come back and share some of the results. True disclosure, Vivian Coe, who I think is on this call, is done the work. She's in the neurosciences graduate program. She came to me just before COVID to join the lab and has done an amazing job of these experiments forward. So these, most of the data and these slides are a result of her graduate work. She's moving through the neurosciences graduate program. So I'm going to go over some background on TDP phosphorylation and specific the kinase family CK1, and then show you the work that we've done in cellular and mouse models trying to dissect out what I think the key questions, and then get to where we're at the end. So let me start with just talking about the heterogeneity of ALS is whatever recognized with chemical and the different phenotypes. Then underlying it is the fundamental aspect, first of all, of motor neuron degeneration, and secondly is the anatomical spread of the disease. And we all know that there's about 90% of the cases are sporadic, and 10% are genetic, and there's a variety of different genes that are responsible for the genetic aspects. But one thing that is common to almost all patients is the TDP-43 pathology. And so about almost 97% of all ALS cases have displayed TDP-43 pathology, and I'll get into that in just a second. And of the abnormal, and that includes most of the genetic causes. The only two that are not related to TDP-43 pathology seem to be the SOD1 and the FUS mutations. But most of the other mutations also, such as C9, are related to TDP-43 proteinopathy. And the TDP molecule was discovered in 2006 in the laboratory of Virginia Lee. And one of the first observations was that most of the rare genetic causes of TDP-43 were located in a very unique domain of the protein, in the G-rich domain, or that was earlier called the prion domain. And that's led to a number of TDP-43 abnormalities listed here, the acetylation aggregation, and specifically today I'm going to focus on the phosphorylation. And this is what we see under immunofluorescence. These are the large motor neurons, and in normal, the green is the TDP-43, and normally it's located in the nucleus. And two things happen in ALS. One is that you lose the nuclear signal, so there's loss of nuclear TDP, and then there's aggregation in the cytoplasm of TDP, and it can organize, it's thought to kind of organize into these puncta that then organize into and then ultimately into dense round inclusions. And it's just another picture from my laboratory showing just what this looks like under immunohistochemistry. See here, first of all, the motor neurons are huge. They're giant cells. These smaller cells that you see in the background here are the normal glia cells. So you can see how giant the motor neurons are. This is the nucleus and the nucleolus, and you can see here staining is the normal TDP. And then again, you get another view of the puncta as they leave the nucleus and then aggregate in the cytoplasm. So the two rival hypotheses, or the two aspects of pathogenesis that are focused on, are whether it's the loss of the nuclear TDP or the aggregation in the cytoplasm that's toxic, or the combination of the two. But one thing that's common to all of it is the phosphorylation of the TDP. And so that led to, in my laboratory, some of the work in my laboratory, we were using that anatomical spread of the disease to find areas which were less degenerated. And then in the human tissues, doing laser capture microdissection to gather these neurons together. It was kind of a forerunner to the single cell seq that's evolved to what it's evolved to now. But at the time, we would pool the motor neurons, laser capture, and then do genomic profiling. And then in one of the studies, we correlated it with the neuropathy. And clearly, the tightest correlation with in gene expression had to do with this gene CSNK1E, which had a tight correlation. So we began to ask the question, why? What is CSNK1E, which is a kinase, CK1 epsilon? And then it led us to a series of questions: is, well, what regulates the phosphorylation of TDP, and who's phosphorylating it? So that led to thoughts about CSNK1E, which led us to this whole family of CSNK1 family, of which there are two tightly correlated kinases, the epsilon and the delta. And there was a literature that we identified having to do with both of them, and we were particularly interested in the epsilon, at least at the beginning. And you can just see here some of the data, and it turns out that these kinases had been particularly studied in the world of circadian rhythms. CSNK1 delta is one of the regulators of the circadian clock. Epsilon seemed to be a gene where it wasn't clear what its purpose was. So then I was particularly keen that maybe ALS was But in the meantime, with careful literature review, it was clear that this question of phosphorylation of TDP and the critical questions is whether the phosphorylation is a driver of the neurodegeneration or is a reaction to try to protect the cell. And you can see here a couple of the key papers that were postulating that actually the phosphorylation was a driver of the neurodegeneration. I'm not going to get into the details of these studies, but all three of them were sort of pointing not only to the role of toxicity of TDP phosphorylation, but specifically about a CK1 dependent pathway. So that's just kind of a simplified cartoon. You see the TDP in the nucleus. What happens in ALS is the mislocalization to the cytoplasm and the phosphorylation of the TDP, and then its aggregation. And the key question again, was it the loss of the nuclear function or the aggregation in the cytoplasm, and was CK1 the key driver? So or was the key regulator? So here's again listed as a question: is CK1 the primary kinase for TDP phosphorylation? After these identifications, is the TDP phosphorylation toxic or protective? It's target. So all of these questions are interrelated, but different aspects of the research. So I want to show you some of the data that we've generated in cellular models related to this. And this is a model that was developed by Haiyang Yu and Don Cleveland's lab, who's here at UCSD. And he's added a component to the protein that then leads to its aggregation in the cytoplasm. So we use this model. You can see it here, that over time you get a mislocalization of the TDP in green, and then you can see the phosphorylation, which comes along with it. This is a phospho-specific antibody, and there's pretty tight correlation with the different antibodies that we have as tools. And you can see here, when in this model, if we try to reduce CK1 epsilon, you can see with an siRNA approach that you can see that it inhibits the phosphorylation of the mislocalized TDP. So this was suggestive evidence that, again, CK1 epsilon was a key regulator of TDP phosphorylation. And then here again, you see in a different, in sushi cells, which is now in a neuronal cell line, again, that either inhibiting CK1 delta and epsilon will inhibit the phosphorylation of the TDP, and epsilon seemed to be a bigger target for delta, at least in this experiment. So in this experiment we took, and so it turned out that there were inhibitors of the CK1 kinases, and I'll come back to that more later when we get into the animal model. But again, we could use the inhibitors, the molecular inhibitors, which are essentially drugs, to test the phosphorylation. Two main kinds of inhibitors that had been developed. One was a CK1E specific inhibitor, and one had mixed actions on both the delta and the epsilon. And this is showing the potency of the drug and leading to very robust inhibition of the phosphorylation with very favorable properties in terms of the potency, which indicated to us the potential of the drug is forward as a drug. Okay, so with that experimental work and some others that we're doing now, it raised the question, well, what does this do in vivo? The cell or one that's important in an in vivo setting. And one comment I would make is that all of the cellular models in ALS do not really model a delayed onset progressive neurodegeneration. So they model various aspects of the biochemistry, but it's hard to come up with a model of progressive neurodegeneration for which we could test using some of our different approaches. So we turned to the mouse models. And again, with the idea that epsilon was more important than delta, at least at the beginning. And let me share that information with you. And the model that we chose is a model that was published in Virginia Lee's laboratory with a doxycycline inducible model of TDP mislocalization. It's a kind of a complicated overexpression model, but essentially what you have is that in the presence of doxycycline, the transgenic activator is inhibited, and so the system is at rest. But when you take the doxycycline off, so the dox is off, then all of a sudden there's expression and then induction of a TDP without a nuclear localization signal. So it drives into the cytoplasm and has progressive cytoplasmic accumulation. And as you can see here, it has not only the biochemical and the histological delayed onset progressive changes, but it causes progressive motor neuron loss, and it has a pretty robust phenotype, which I'll show you in a second. So we turn to this model, and to be clear, there is phosphorylation of these aggregation in the cytoplasm. It mimicked a lot of the important features of the human disease. Okay, and this is the work that we did with the model to just recapitulate what had been published, showing that again, when the doxycycline is stopped, there's induction of the transgene, accumulation in the cytoplasm, progressive phenotype, and you can see there's forearm grip strength, rotor rod, compound muscle action potentials, and there's even a marker rise of the neurofilament light chain. So it and progressive mortality. So you can see that it's at least from my point of view, it's a nice model for a delayed onset progressive neurodegeneration with related phosphorylation. Another aspect that I like about this model is that, well, let me go to the next slide. And so it turns out that there was a mouse model that had a knockout of the CSNK1E. That's the CSNK1E is the gene, and then the protein is CK1 epsilon. And it turns out that the mouse model really has no phenotype, and it's well tolerated. So you can knock out the CK1 epsilon gene without any consequences. The delta gene, just to be clear, you can't knock out because those are embryonic lethal. So it's not a model that could really test easily anything related to the delta form of the CK1, but it would lend itself well to testing about CK1 epsilon. And it's a complicated breeding strategy, which was supported by Don Cleveland and his veterinarian to help us work out this model. But we figured if we could breed the knockout CK1 epsilon with the TDP model that I just showed you, we could test the hypothesis that epsilon was really the primary CK1 that was involved in TDP phosphorylation. And you can see here in the knockout, we had good knockout of the CK1 of the gene. You can see with relatively sparing of the CK1 delta. Okay, then, so we did the experiment, and we were disappointed, to be candid, because what happened was that when we compared the wild type with the knockout, it wasn't a clear impact. We were predicting a major sparing of the phenotype, and we just didn't see it. We tried it at one point, trying to make something favorable out of that, but at the end of the day, it really, looking out CK1 epsilon did not have any major impact on the progressive neurodegeneration. But then Vivian did look at tissues to see whether or not we had really inhibited the phosphorylation, which was our initial question in the first place. And you can see here that in the knockout, phosphorylation persisted. So even though there was no CK1 epsilon, something else was modulating the phosphorylation, separate or different than what we were seeing in the cellular models. So this led us more closely at this delta epsilon. And so the project morphed at this point into a more close attention to the interaction between the two isotopes, or the two different forms of CK1, the epsilon and the delta. And we had already were well aware that the inhibitors could inhibit either epsilon, they were epsilon specific, or they had action on both epsilon and delta. So we turned our attention next then to the pharmacological approach. The genetic approach clearly, I think, showed that the epsilon itself was not sufficient to drive the phosphorylation. So we couldn't really test whether inhibiting it with epsilon inhibition. But we had these inhibitors, which I've already shown you, had this high potency, and they could be either epsilon specific or epsilon delta specific. And so we became more enthusiastic about these inhibitors, one of which, compound 9, is called, also we know it as compound 9. By the way, I should say that as we were inhibitors had been developed by Pfizer, that's why they're called PF initially, but then they were sold to Biogen. And so we contacted the research team at Biogen and fortunate to work closely with them. We've learned a lot of medicinal chemistry, and we are working closely in terms of the drug development, even though they have all of the intellectual property and all of the mature inhibitors. It's been a great partnership because we have industry elite medicinal chemists to advise us and work with us on the projects. You can see here that, again, the specificity of the compounds, this just shows that this is a kinase, kind of an assay showing which kinases, and it shows that these compounds are primarily specific for the CK1 epsilon. This one is epsilon delta. As you get to higher dosing, which is an issue we're going to have to deal with later, is you lose some of the specificity. So that's in the future, we'll have to try to define that better. But the first question is, do the CK1 inhibitors reduce the phosphorylation in this mouse model? And so here you can see there are two questions really to do it. Is by inhibiting with the different inhibitors, do it in vivo. And secondly, what's the time course of the induction of the phosphorylation in this mouse model, which we had to dissect out. So you can see the progressive, after the induction of the transgene, you can see progressive phosphorylation of the TDP starting at about day 5. This is long before, not long, but it's before the mice develop symptoms. So it's a prodromal to the phenotypes or the behavioral changes that come later in the induction of the gene. So we were able to define the time course and then to look at the inhibitors to see how effective they were in inhibiting the phosphorylation, now that we could inhibit both delta and epsilon simultaneously. And so this is kind of where we're at this point. So the phosphorylations can be reduced in the mouse model with compound 9, which is the compound that I said had the action on delta. And here you can see, beginning to see that it's a dose-related decrease here in compound 9. Well, here you can see that the progressive decrease of the hyperphosphorylation. You can also see a rise in the, a temporary rise in the neurofilaments. We're trying to sort this out, but you can see the effect on the neurofilaments, and also having to do measuring the levels in the plasma of the mice and in the brain. And then looking at the ratio of the penetrance of the compound in the nervous. And again, showing that we think with the levels that can be reduced with compound 9, and one of the things that we're now calibrating is the different routes of administration. And so one of the things we learned in the mouse models is that can give it subcutaneously, but to have an effect, a medicinal effect, that's a lot of work for the mice, for the repeated injections. We could do IP dosing, and then ultimately you can actually formulate the chow so that it's in their diet. So what we've evolved to now, trying routes of administration, and with the potential impact on the animals as for the experiment, is trying to figure out a chow formulation so that we can deliver the compound, the inhibitor, to the mice with minimal intervention. Relation of the TDP, and you can see that in one case there was minimal inhibition, and in another case there seemed to be almost complete inhibition of the TDP phosphorylation. So this led us to feel vulnerable in the experiment, as if how could we predict who was getting the compound, and that's what we think happened in this mouse case, is that the drug wasn't delivered, and in this case it was. And so if we're really going to test the hypothesis, we have to have confident, reliable delivery into the CNS. And so that's led us to the chow formulation. And so here are the preliminary results as we're moving forward with this. This is going to be where we're going to end the our experiments for this presentation. But you can see here again, looking at the placebos in red, treatment is in blue. This is with the chow formulation at a relative dose, and you can get a hint improvement of the phenotype. This is looking at grip strength, forelimb grip strength. This is looking at rotor rod. This is looking at the compound muscle action potential, and then look at body mass. But you can see at least hints of efficacy, at least in a lot of the metrics, that we think that those that are receiving the compound 9 inhibitor, so both epsilon and delta inhibition, are having what are suggested to be improvements. One of the things we're trying to calibrate now is the dosing of the chow formulation with the degree of inhibition of the phosphorylation histologically and biochemically. So those are where we're at now. We're poised to try to do some dose calibrations of the chow so that we can get optimal inhibition of the phosphorylation, and in turn try to determine whether it really does have a significant impact on the phenotype. If it does, then we have, I think, done a lot for proof of principle that the phosphorylation, in fact, is a participant in the toxicity and the neurodegeneration. So we're particularly excited in approaching the problem in this format. So I'm going to stop here and just summarize and say that the TDP-43 phosphorylation is a primary pathologic feature of ALS, but it's still not known whether this is a primary toxicity or a secondary protective mechanism. Delta and epsilon are upstream regulators of TDP phosphorylation. Reduction of CK1 delta and epsilon expression or inhibiting the kinase activity significantly reduces phosphorylated TDP-43 levels in cellular models. The CSNK1E knockout studies, along with the studies in mice, have suggested that inhibiting both the delta and epsilon is needed to reduce phospho-TDP levels. Inhibiting CK1 alone sufficient in vivo. And then engagement of mechanism for the delta epsilon inhibitors is established and does reduce TDP phosphorylation in mice. And so now we're doing the proof of principle, we're trying to establish proof of principle or test whether CK1 delta and epsilon inhibitors are protective or toxic in vivo. And these results, I think, would be very exciting if they're positive, and even if they're negative, it would teach us something about the role of phosphorylation, which still remains enigmatic. So either way, with these experiments as we proceed, we're excited to drive it forward to the conclusion. And I've already shouted out for Vivian, who's done this work, and her team, and other people in my laboratory. A shout out to Steve Wagner, who advised us a lot on the medicinal chemistry in the early days, and unfortunately recently died, and we've been sad about that. And then a lot of our collaborators, both at UCSD and Biogen in particular, we've been partnered with, and they've helped us. And of course, the FightMND has really been the main primary support for these experiments. So thanks to FightMND for all of that.
Great. Thank you so much, John, for your presentation. We'll be opening the floor up for questions. If you have a question, you can either raise your hand and we can activate your camera and microphone, or if you'd like, you could pop your question into the chat. We just ask that if you are going to ask a question, to please introduce yourself, your name and your affiliation, so that we can all get to know each other a bit better. While we're waiting for people to jump in, I might jump in with a question if that's okay, John. Really interested in your data with the CPD compound and the reduction in the phospho TDP-43. And I was wondering if you've got experiments planned to look at whether or not there's a reduction in TDP aggregation and motor neuron loss, and also looking at neuroinflammation as well.
Yeah, good questions. So we've been kind of laser focused on the phosphorylation component. I think the model does have aggregation, and so certainly as we tease apart the biochemical aspect of the phosphor and histological, we'll be definitely looking at the aggregation in this model. And yes, there's a lot of talk about the inflammation, and I think a lot of how far we're going to take this will really depend on whether there's a major effect on the phenotype. That's what we're really after. Is this inhibiting? Does this have a behavioral effect on the disease progression?
Thank you. And one other sort of a bit left of center question is, I know you were saying that the CK1 delta can affect circadian rhythm, and have you looked at that in these mice with that inhibitor?
Yeah, great question. I don't think it's been a problem so far. We're at the low doses of the drug, and I think the concern, and I believe that the delta inhibitor has been tested in humans, and this has been looked at for Parkinson's disease and other neurodegenerations. And so there's definite concern about what inhibiting delta is going to do. So we're extremely mindful of that. I think the first and foremost is the biochemical question, independent of toxicity and effective circadian rhythm. Is that sufficient to really impact disease progression? So once we get that, there'll be lots of follow-on studies about the circadian disturbance.
So to the floor, any questions? Yes, Julian, I'll just allow your microphone, and if you wouldn't mind turning on your camera and introducing yourself.
Okay, thanks, Beck. Hello, John, and thank you very much for an excellent talk. Julian Gold's my name, and I'm CI of the Lighthouse 2 project, which you know very well because you're on the steering committee. Just a sort of a tangential question, because as you know, my interest is in HERV-K. And there's some suggestion that if you look at the enzyme asparaginyl endopeptidase, which is supposed to cleave TDP-43, and there's some suggestion now that that HERV-K downregulates this enzyme and therefore allows TDP-43 to accumulate. I'm just wondering how do we reconcile that with your work, and if we're doing this multi-centre trial looking at Triamec, which is going to suppress HERV-K, and we know it does that. Are there investigations that we should be doing on that population? Because it's a large population, as you know, in the UK and Australia and New Zealand and Europe, to support this work that you're doing.
Yeah, so fair questions. Let me make sure I'm addressing it right. But I mean, I don't see them as well, I think one of the questions I have about the HERV-K story is whether that's upstream or downstream of the TDP. I mean, as you know, that's some of the endogenous retroviruses. The questions are those released from the neurodegeneration, or are they upstream driving the neurodegeneration? So I think related to the phosphorylation, I see as which is upstream of which one is upstream. And if HERV-K is the reduction releases, what am I trying to say? So that if abnormalities of HERV-K lead to abnormalities of TDP, which then in turn translocate and accumulate, then that would put the phosphorylation pathway further down and a protective mechanism. That's what I see is happening a lot in the ALS field right now, is all these different pathways: autophagy, mitophagy, axonal transport. Those are all downstream pathways. So how far upstream can we get? And I think what we're excited about or working on is whether or not the phosphorylation is whether there's a primary regulation of that, and that becomes imbalanced and leads to TDP abnormalities, or will it salvage anything, or is it really all just protection?
Okay, thank you. Well, we'll know more when when we do the clinical work.
Yeah, there's no way now in patients, as clinical trials going on that I'm aware of. The neurofilaments is a biomarker, and that's a useful biomarker, but it's a direct readout of phosphorylation. I'm not aware of anything that's available that could, as data that could be gathered, except pathologic. It's all, the phosphorylation is really a pathologic story.
Okay, thanks very much. Thank you.
Yeah. You.
Thanks, Julian. And we have a question from Simon Maxua. You should be able to turn on your camera and microphone. Hi, Simon. Your camera and microphone's been enabled? Yes, you can ask the question in the chat, no problem.
Beck, I've got a question while we're waiting, if that's okay. Oh, here it comes. I'll go later.
Okay, thanks, Darwell. So the question is, in regards to the functional data, you see a decline in rotarod. Do you think additional dosage would help improve this function?
Yes, is the immediate answer. I think these were pilot studies, and with a pretty low dose. And when we've done the biochemistry, what we're seeing is that there's still a lot of phosphorylated TDP. So we haven't sufficiently inhibited the phosphorylation, yet we're already seeing what we think is a positive phenotype. So we're definitely going to try to scale that up to as high a dose. So we're dosing it. That was at 20, and we're going to go to test 40, 80, and 120. And then once we get the dose calibrated, assuming there's no toxicity, the other thing we have to obviously protect against is the toxicity. Once we get to the optimal dose, then we will do the prospective study after dose optimization. So definitely we're going to push that as high as we can.
And this follow-up question, would multiple doses be an option?
Well, okay, so that's why we're switching to the chow. So I've always maintained that a lot of the mouse experiments seem to be done with single injections or daily injections. A lot of the ASO trials for the gene therapies have been done with a single intraventricular injection. And that's not the way we treat patients. Patients are, you have a steady state drug exposure, and you dose it, once a day if possible, but twice a day up to 4 times a day, because you want that steady state. So, and if we're really going to test whether the phosphorylation is involved with the toxicity, we need to suppress that, not episodically, but continually. So that's what led us to the chow formulation, because we think we can then achieve a steady state of drug and of phosphorylation inhibition. Absolutely.
Great, thank you. And just some feedback, great talk, exciting work from Simon, so thank you. Davo, I've got a question for you from you, and then Corey will throw to you next.
Yeah, hi again, John. Fantastic talk, and thanks for being involved in the seminar series. Just looking at the question about the behavioural and functional readouts, which the grip strength seemed to be very, very impressive. I gather you're looking at some lifespan experiments going forward as well, in terms of increasing the length of life of the animal models, or are you looking into doing those kinds of experiments and also moving into additional models involving the TDP aggregation?
Okay, so I missed the key word there. So are we looking at what?
Are you looking at lifespan? Like, have you done any experiments to lifespan? Yes, and longevity.
Yes, the answer is yes. So what we'll be doing in the big experiment is, once we get the dose calibrated, is that will be a huge experiment. And then what we will do is be looking at all the behavioral data. We'll be looking at electrophysiologic data like compound muscle action potential. I stayed away from some of the details of that. We'll be looking at neurofilaments and so on. And then we'll be looking at lifespan, and then we'll be sampling to look at all the biochemistry and the histology. So to do that, we have like parallel cohorts, some of which are destined for sacrifice for the biochemistry, for example, at different time points. And I think one of the things we're building into the model is that at some point when the mice look sick, we'll call, there's criteria for whether they're, dead or not, and then we'll be looking at the Kaplan-Meier mortality curves, survival, but at the same time trying to get the mice for other measurements. So yes, that'll be. In terms of taking it into other models, I'm not sure where it's going to go next. I think if we have a really robust readout, which, obviously everybody wants, or we want, but I mean, we were careful to, first of all, say we're testing a hypothesis, so we're not committed to the outcome. We don't want to drive forward something that is not having a significant impact. There is one other mouse model of TDP, which I'm pretty sure has phosphorylated TDP, that was used for the ataxin-2 studies. So I think the follow-on, if there's some significant proof of principle from this, is that will be carried on into further studies. Ultimately, what we want to do is get the compounds into a human trial, but we've got to have enough proof of principle that that's worth doing. And that's our goal for this grant.
Thanks, John. I look forward to the outcomes going forward.
Thanks, Davor. Corey Holdham, your microphone and camera's been enabled.
Hello. Thank you for the talk, John. It was quite enjoyable. I'm sorry I don't have a camera right now on this machine. I'm actually asking this question on behalf of Shuno, who was here, but she's dropped from the call. So she's passed me a question to ask you. But based on the results that you were showing earlier, it looks like there was compensation from the delta, as the knockout of epsilon alone had no effect. So how do you plan to titrate both in combination? And then in addition to that, titration with combination with other drugs like edaravone or riluzole.
Okay, so first of all, I think what we're hoping for with the inhibitors is that there'll be simultaneous action on both. The inhibitors are type 1 inhibitors, so they fit into the kinase pocket and then prevent it from phosphorylating the TDP. And so we are trying to dissect out whether there's compensation between epsilon and delta, so that when you inhibit epsilon, for instance, there's a rise in delta. So we're trying to be mindful of that interaction. So that's one aspect. One thing to say is that these are kinases. So one thing I'm learning is that it's really not just levels of the protein or of the expression, it's really the action. So trying to look at, are we inhibiting the action rather than just the levels of the kinase? So kinases are a little more complicated in terms of functional readouts. But ultimately, I think the inhibitor inhibits both epsilon and delta simultaneously. So that's the way we're going to try to move that forward. I think that answers your question.
Yeah, thank you.
Thanks, Corey. And John, we've got one more question in the chat, which is from Lorenzo. So Lorenzo is a postdoc in Kathy Blizzard's group at the University of Tasmania. Their question is, I'm curious about how you plan to find out whether phospho-TDP is toxic or not, and that surely phosphorylation needs to be a dynamic process, so suppressing it might be just as detrimental as if it's enhanced. And do you think we can use pharmacology to understand mechanisms, or other approaches, better approaches needed?
Good, great questions. Let me just think about that. So, we tried to, yeah, for sure. I mean, phosphorylation is a dynamic process, but yes, and by inhibiting assuming that we can inhibit the phosphorylation, I guess that's one thing. Is, are there other kinases, and then what are the off-target effects? And then what are the consequences of doing it in of inhibiting in another tissue? So it definitely leads to a lot of complexity. I think still the simple question is using pharmacology to try to understand the physiologic mechanism. So we don't know whether the phosphorylation is protective or toxic. It's hard to study it any further from a genetic approach, at least in mouse models. In cellular models, we're trying with ASOs and siRNA. We're trying to look at that in cellular models. But as I tried to indicate, the cellular models don't have a delayed onset progressive neurodegeneration. So that's where the animal models come in. And we are trying to use a pharmacologic approach to dissect a physiologic mechanism, but of course it works the other way around. In pharmacology, it goes both ways. But for the simple-minded question from my point of view, which began as a clinician, is there a drug and can we ameliorate the progression of disease? And that led to a simple question about phosphorylation. Will inhibiting that help or not?
Great, thanks. And great that you have a drug that's blood-brain barrier permeable as well, which doesn't come very often.
Right.
Great. Thank you, Sarah. Sorry, John.
Yeah, no, I was just going to say, I think my understanding, I'm new to the field of kinase biology and pharmacology, but reading through the literature and talking to people, there's been kind of a revolution in how kinases may be brought into the clinic for therapy. So it's a new world.
Great. Thank you. Over to you, Sarah.
Thank you very much. Thank you very much, John, for a fantastic presentation and a really engaging discussion session. If you have any feedback or would like to follow up with our speaker, please do get in touch with me at FightMND and we can connect you in collaboration. I'd like to engage. So today's seminar has been recorded and will be available on our website in about a week. So if you do know anyone of your colleagues who weren't able to join in live today, let them know that they can watch it soon on our website. So again, thank you, John, for taking the time to speak about what means today. We'll see you next time at our future seminars, and that's all for today. Thanks.
And thank you.
Yeah, thanks, John. That was a really great presentation. I really hope everyone. Well,
Thanks
we
again.
do have quite a few people sort of watching it. Bye.
Okay, talk to you later. Bye-bye. Thank you.
Thanks so much, John. See you later.
He's gone.